So Unit 1, Chapter 1 of System Neuroscience. Let's talk about the organization of the nervous system first. You can divide it into central nervous system and the peripheral nervous system. So the central nervous system is made of brain and spinal cord, and the peripheral nervous system is made of the nerve fibers and also ganglia. Ganglia are collection of neuron cell bodies that are outside of CNS. Functionally, it's a little bit different, okay, the organization. So you have the afferent system and you have the efferent system. So these two belong to peripheral nervous system, and they connect peripheral nervous system to the central nervous system. So part of the afferent and efferent divisions are also inside of the CNS. Let's talk about the afferent system. So it needs receptors. Receptors are like receivers. They are stimulated by different type of stimuli, and then they are activated and they are sending information to central nervous system, and that's why they're called afferent. So there are three different type of receptor. One is called the special senses. So this is the sense of smell, taste, vision, balance, and hearing. There are also visceral senses, and they are in the organs. So most of the time we are not aware of it, but it can come to consciousness, and sometimes we can feel it. For example, increasing heartbeat, or if you're breathing more, or your stomach is growling, so you can feel that. And it's the somatic. Somatic is the conscious one. So it's most of the time it's in the skin, but you also have it in the skeletal muscle, in the joints, and in the tendons. So we will come back to that later on. Okay, so it's a different chapter. We will talk about it. So receptors are here. They are going toward the CNS. They enter the CNS. So it can be in the spinal cord or in the brain itself. Depends on the location. The brain now processes sensory information. It's involved in a higher brain function. For example, judgment, planning, emotions, memory, learning, all of these behavior. So there is different functions there. And the motor response. So any sensory stimuli that you get, you may or may not get a motor response, depends on the type of stimulus. So now the motor response is leaving CNS. So this is why it's called efferent. So the efferent division is motor, and you can divide it into two parts. One is the somatic nervous system, and the other one is autonomic. So this somatic nervous system is not the same as this one. Okay. So soma means body, and here is that it is going to the effectors. So effectors are anything that the nerves are innervating. So in this case, it's skeletal muscle. So it's under your conscious control, and you can move any part of the body when you need it. So then is the autonomic nervous system. The autonomic is not the same as the visceral. So this is visceral sensory. The autonomic here, although it means it is subconscious, but in most sources that you see, it is under motor system, not under sensory. Okay, so it's a little bit confusing, but autonomic nervous system is mainly motor. And that divides into parasympathetic and sympathetic, and they are innervating smooth muscle, cardiac muscle, glands, adipose tissue, and immune tissue. Okay, and the balance of these two divisions are important for homeostasis. Now look at the location of different nuclei or ganglia. So if the collection of the cell bodies are inside the CNS is called nuclei, outside is called ganglia. So for the peripheral nervous system, you can see the sensory and cell body bodies, sensory neurons, cell bodies are outside of the CNS. So these are the special senses, and this is the general senses, and the cell bodies are outside, and then they either enter the brain or in the spinal cord. There is more detail to this, so don't worry about it for now, just to know that the cell bodies are located outside of the CNS. Then they enter, and then they go and they are processed in different parts of the brain. We will talk about that later. And then the motor system. So the motor system, the first cell body is located inside the CNS, the first one. And if it is an autonomic nervous system, autonomic one, so you have another cell body outside of CNS. So you see this ganglion is a collection of neurons outside of CNS. So you should have another one here too. So this is outside of CNS, so for autonomic. But for the somatic one that is conscious, then you do not have another cell body here. So that's the exception here. So for the sensory, you have the cell bodies outside. For the motor, you may or may not have it. So if it is autonomic, you have another cell body outside of CNS. If it is the somatic one, you do not have it. This is another way to divide the nervous system. So again, the central nervous system and the peripheral nervous system. So you can divide it into somatic. So somatic is conscious. It's all the sensory information. So that can be general sensory, for example, pain and temperature and touch. Or it can be special senses, for example, sense of vision and hearing and balance, things like that. And you have the motor one, which is the skeletal muscle, so it's under conscious control. Then autonomic nervous system. So this is the thing, is the source that I took it from this book. I don't think this is the way that they labeled is very accurate, because autonomic nervous system is mainly motor. Okay, so this should have been a visceral nervous system. So the visceral nervous system can be sensory, so that is a subconscious, or it can be motor, which is the sympathetic and parasympathetic nervous system. And there is another one is called the enteric nervous system. So enteric, again, it is related to gastrointestinal system, and technically, if you think about it, it's one of the visceral organs, some of the visceral organs, so it should be under autonomic nervous system, but it is separate. Why? Because the gastrointestinal system, they have their own nerves, and most of the time they are controlled locally. So anything that you eat depends on the content of that. So there are different enzymes are released, and different hormones are released, and also different activities that are happening, absorption and secretions. So most of the time they are local, but despite of that, they are controlled by the autonomic nervous system. So the movements, the peristaltic movements, the secretions, are also controlled by autonomic nervous system. So when we get to the visceral nervous system, I will show you this idea that there are some neurons that are locally located in the gastrointestinal system. There's also some neurons are located, or cells that are highly active, and they can send electrical potential. They are in the heart as well. That's the electrical conductive system of the heart, but they do not separate that. They are still under the autonomic nervous system. So it's a little bit a naming difference probably. So the histology of the nervous system. So the nervous system is divided into the neurons, okay, and neuroglia, or glial cells. So the neurons are the ones that they are responsible for electrical activities, sending and receiving information. And neuroglial cells are supporting cells, and most of the time they may have some electrical activity, some of them, but mainly it is for support. So the neurons are made of three different parts. Okay, this is the dendrites, this is the cell body, and this is the axon. So dendrites receive information. Okay, dendrites are receiving information. So you have you have channels here, and that are activated when the chemicals are released, and they are stimulating different channels here. So then they send information to the cell body. So cell body of the neurons are similar to other cells, but they are very, very active. They have a lot of the ribosomes, so rough endoplasmic reticulum is very active here. And when you look at it under microscope, it looks darker. So if it looks darker, that is because of the high number of the ribosomes in the endoplasmic reticulum. So these are called nestle bodies. You have a lot of mitochondria also, so aerobic metabolism is there, and you have a lot of different cytoskeletal, so microtubule is a good example there. So the cell body is processing information also. It makes different chemicals, the neurotransmitters, and they can send it down in this long extension. This area is called axon hillock. So this is the area that connects the cell body to the rest of the axon. Okay, axon is the one that sends information. So it's a one-way direction. So dendrites receive, cell body process, and axon sends information. And you can see that in some areas the axons split. So these are called the collaterals. And at the end of these collaterals, then it branches again to a smaller one. These are called the axon terminals, and at the end there is a bulb, so an area enlarges, and that is called the synaptic bulb or synaptic terminal. Well, when the chemicals that are made in the cell body that are moved through the axon, so proximal to distal, this is called anterograde transport. But also the neurotransmitters that are released here, they can be picked up. Some of them are recycled, so they are made here. Some of them are going back all the way to cell body, and that is called the retrograde transport. And retrograde transport is also important for formation or development of some of the diseases or abnormalities in the neurons. Some neurons are covered by these neuroglial cells. So example here is the Schwann cells. Schwann cell is in the peripheral nervous system, but the central nervous system also is covered by different type of cell. Not all of them, I mean by different type of cell. I meant not Schwann cell, is called oligodendrocyte. I will come back to that. But if you look at it, it's this area, you know, it's covered, so this area that is covered, this is called myelinated part of the neuron of the axon, and some area that are not covered. So these areas are not covered. So the area that is not myelinated is called the node of Ranvier. So there is an importance to that for electrical activity, and I will show it to you later. So types of neurons, so we're still in the histology of the neurons. One is called anaxonic. It means there is no axon here. You have a lot of dendrites, and you can see that in the brain and also in the special senses, for example in the eye. And bipolar neuron, it means has two poles here. One is the dendrite receiving information, the cell body process information, and then it's a short axon that sends information, and you see that in the special senses. Again, for example, in the retina of the eye, you can see it in the nose in the sense of smell, and also you can have in the ear. And these ones are really small. Okay, so the length of these ones are really small. Unipolar or pseudo-unipolar are the ones that are they have one cell body, one area is the dendrite that receive information, and then is the axon that send information. So from here to here all the way is the axon. The difference between this cell body and this cell body is that this one processes information, this one does not. This is just sending information. And then you have the dendrites, I mean the axons that are branched, and then that is activating another neuron and another cell, and that is called interneuron, so inside the central nervous system. And this cell bodies, by the way, is located outside of the CNS, okay, outside of the CNS, and this is in the sensory nervous system. So for example, if this area is your big toe, this will be located near spinal cord. So just imagine this axon is that long, then this part will enter the spinal cord, and then this one is connected to another neuron. It's called interneuron. Okay. Then is the multipolar, so it has many poles here. So they have made of many dendrites and also a long axon, and this is you see that inside the CNS, and also in an area between connection between central nervous system and the muscles, so skeletal muscles. That's called the neuromuscular area. So that is the one of the common neurons are this one. Synapse is connection between the two neurons or one neuron with another cell, another effector. So here in your textbook is shown three different types of neuronal synapses. So it's connection between two neurons, neuronal. So this is the axodendritic synapse. This is the most common. So the axon is connected with a dendrite here, so most common but not strong. The second one is called axosomatic, so the axon is connected to the cell body, so less common but stronger. And the axonal, this is not as common, but this is the strongest because the axon is responsible for sending electrical activity, action potential. So that's why you're directly affecting it. In these two cases indirectly, but here is the direct effect. So this is the strongest effect on the movement of electrical activity. So now in this slide you can see that you have the neuron but also the supporting cells of neuroglia. So these are the neurons, they are really big, and then you have some other cells that are neuroglia. So these are called astroglia or astrocytes. They have a lot of extensions, so astro means star. And you have the oligodendrocytes that are covering the axons, and they do not have as much extensions as the astroglia. Astroglia are, you can look at this, connected to the blood vessels. You have the microglia, they are really small and they are like macrophages. They have ependymal cells, and this is the simple cuboidal cells, and polydendrocytes, which is the stem cells. And neuropil is the area that contains a lot of fiber. So these are the cells, and then lots of fibers of the neuron. These are called neuropil. So let's talk about each neuroglial cell separately. So the first one is called astroglia or astrocytes, and they are divided into the fibrous, which you can see that in the or around fibers of the axon, fibers of the neurons, which are the axons. So they are called fibrous ones. So that's in the white area of the central nervous system. White area is called. And the other one is called the protoplasmic astrocytes, that they are surrounding the cell body of the neurons, and they are in the gray area. So once again, the fibrous one is in the white area, and that are near the fibers, and fibers of the neurons or the axons of the neurons, so same thing. And the protoplasmic ones are in the gray area, which is around the cell bodies. The Mueller cells are the special cells, and you can see that in the retina of the eye, and they are important for homeostasis and metabolism of the retinal tissue. It supports neurons and nurture neurons. Why? Because of the astrocytes are wrapped around the blood vessels. So this is a blood vessel, this one that you see, and this is made of the simple squamous epithelium, so one layer only, and they have these tight junctions, and in tight junctions means not everything can pass through it, and only the lipid soluble molecules, including the oxygen and carbon dioxide, can pass through that. Also water can also pass through it, but not as much. And astrocytes are wrapping around these blood vessels. So the astrocytes are controlling how much material from the blood can go to the neural tissue. So this area is the neural tissue or intercellular area. So this area, so the content of the intercellular area is of the nervous system is very, very different from the content of the blood, and the reason for that is that the neurons are very sensitive to different chemicals, so you don't want to stimulate them too much. Okay, so that way it is important for providing nutrition, but also supporting them as well, so it's protecting them against different chemicals. So this area that is connecting the the simple squamous epithelium and the area that is surrounded by the astrocytes, so this area, this is called the blood-brain barrier. So the blood-brain barrier is like a wall that's separating the blood from the neural tissue. It also has the neurotransmitter function, so astrocytes can convert glutamate, which is a neurotransmitter, to glutamine, so that's another one. And the tripartite synapse is that you have two neurons connecting to each other, and now you have the astrocytes that also is there, and the astrocytes, they have the receptors for different neurotransmitters, but also can release some of the neurotransmitters, so they are affecting the electrical activity between the two neurons in the synaptic area. And the radial glia is that astrocytes during the development of the nervous system, helping the direction and the movement of the axon, so it's connecting different areas of the central nervous system, so that is called the radial glia. Oligodendrocytes and the Schwann cell, they are very, very similar. Okay, so this is why I explained them together. The oligodendrocytes or oligodendria they are in the central nervous system, and Schwann cell are neurolemma cells, and they are in the peripheral nervous system. Okay, they both wrap the axon. Okay, when they wrap it, and that is called myelinated neuron. So that area that is covered is myelinated neuron, and that actually protect the axon from being damaged. Okay. Also, it's important for the distribution of ion channels. Why? Because in this area there is myelination is not there, so ion channels are located here in the node of Ranvier. An area of the axon that is not covered by the myelin sheet is called the node of Ranvier, and the ion channels are located there, so it's important for the amount of the ions that pass through from inside and outside of the cell, so it's affecting that. So it's important for the action potential. And also then in these areas between the nerve and the muscle and the Schwann cells, it can pick up extra neurotransmitters, so that the extra neurotransmitters that are picked up, it can be recycled. Okay. If you look at this image, you can see this is the axon, and this thing that you see around the axon, this is called axolemma. So this is the plasma membrane of the axon itself. Axolemma, the plasma membrane. Then this is wrapped like a hot dog, okay, around the axon. So this whole thing is the Schwann cell, and the Schwann cell has these layers, and that layers is made of fat, and that's called the myelin sheath. So the axons that are myelinated, they have a higher speed of conduction. Microglia, they are small, they have a lot of branches here, and they are like macrophages, and if there is any debris, cell damage, any pathogens, they come and they remove them. So this is why they are important for the inflammatory response. Ependymal cells are the simple cuboidal cells, and they have a lot of cilia. In some area they make a choroid plexus. I will come back to that, don't worry about it. So later on in a different chapter I will discuss this in more details. And they produce a cerebrospinal fluid. Cerebrospinal fluid, they are in the cavities, okay, in the cavities in the brain and the spinal cord. Also the brain and spinal cord floats in the CSF. And the polydendrocytes, these are the cells that are stem cells, and they can convert to either neuron, or it can convert to neuroglia, most of the time to neuroglia, because most of the time the neurons they don't divide, and there's some exceptions there, but most of the time they don't divide. So that's why if there is damage, the damage is permanent. So now is a different topic, neurophysiology. So this is, we are talking about the membrane potential. If there is no stimulus there, this is called resting membrane potential. Resting membrane potential, no stimulus. Okay. And I will come back to the resting membrane potential. What is it made of? And then is the graded potential. So the graded potential, it means there are some changes in the membrane potential. Okay, so once these dendrites are stimulated, or once the cell body or the axon hillock is stimulated, also here in the postsynaptic area of some of the cells, when there is a change in the membrane potential becoming more positive, that is called graded potential. Graded potential can also be negative, okay, but most of the time it's positive, but it can be negative. So then is the action potential itself, that is mainly, you see it in the axon, okay, so it transmits long distances and it will not decay, so it does not decrease. And so one area is the axon of the neuron, and also you can see it in the postsynaptic membrane. For example, in the muscles you can see that. So in the neurons, axon, and also in the plasma membrane of the muscles, that you can see an action potential. So here is the cell, and you can see that this is the extracellular fluid, this is the plasma membrane and the cytosol. So the plasma membrane is separating the two areas, so the content is different, because that is a semi-permeable membrane, if you guys remember it. There is different ions outside of the cell, so the sodium is responsible for positive charges and chloride is positive for the negative charges. Inside the cell, in the intracellular fluid, you have the potassium that are positively charged and the proteins that are negatively charged. Okay. Then you have these channels. So you have two different types of channel. One is called gated channel, which is not shown here, and the other one is called leaky channel, which is shown here. So for the leaky channel, the one that are important is the sodium and potassium channels. So you can see that here in the potassium channel, the potassium is leaving the cell. Potassium is leaving the cell. Okay, so and the sodium likes to enter the cell, because there is not enough potassium outside of the cell and there is not enough sodium inside of the cell. but you have this sodium potassium pump or ATPase pump, and this is the one that brings 2 potassium in and kicks out 3 sodiums out of the cell, and that requires an ATP. This is why inside of the cell is more negative and outside of the cell is more positive. So inside the cell, the resting membrane potential is around 70. It can be 75, 65, but roughly around negative 70. Okay. And the reason is that you have more proteins here, so the proteins are big ones, right? And they cannot pass through these channels, so they have to stay here. So if the potassium is leaving, you lose some charges, okay? And if the sodium comes in, then it's kicked out by the potassium, so that's why it's negatively charged, and that is at rest, okay, not during stimulation. During stimulation other things will happen, but happens at rest, and it's always there. These channels are passive channels, they do not require any ATPs. So here there is two different areas that is separated by a semi-permeable membrane. So here the content of the potassium is high, so it moves from a high or more concentrated area to a less concentrated area until it balances out. So we are talking about one ion at a time, so it will be at a time that everything on both sides will balance out. If it is a sodium, so this will be outside of the cell, it will move inside of the cell until it balances out. We are talking about individual ions, okay, not all ions. So if you allow this, everything will be balanced out, and what will happen then, that will be called equilibrium potential or electrochemical equilibrium. So it means on both sides is the same. But in reality it's not like that. So if it is you have a lot of potassium inside the cell, you can move this potassium out, so this is the concentration gradient. But also you may have the sodium. The sodiums are positively charged here, so you do not the similar charges will repel each other. So this is why if you have too much of the potassium, it will not allow more potassium to enter, because you have the sodium, so that will repel, and that is the electrical gradient. For potassium you have more concentration gradient than the electrical gradient, but the sodium is not shown here. So the sodium is positively charged, and it can enter the cell more because the cell is more negatively charged. So for the sodium you have more electrical and chemical gradient, but again you have the sodium potassium pump, and the sodium potassium pump will kick out the extra sodium, so the extra sodium will not be stay inside the cell. But if we go back to the equilibrium potential, for the potassium is negative 90, and for the sodium, if you allow the sodium to enter the cell, and then it will be positive 66 millivolt. But as you know, inside the cell, the resting membrane potential is around negative 70, so it's a lot closer to the potassium than it is to sodium. And one of the reasons is that the potassium leaky channels are 25 times leakier than the sodium channels, 25 times leakier. So that's why the potassium can leave the cell, making it more negative. Okay. And then you have the ion channels. You have these are the gated channels. So you have to have a different stimuli to activate these channels. So one is called a voltage gated channel. So the voltage has to change here, and that will activate this channel, and ion can enter the cell. So this, for example, you can see that in the axon. The other one is called ligand gated channels. So you have the chemicals there, and these chemicals attach directly or indirectly to the channels, and then they will open up the channels, and then the ion can enter. So you can see this, for example, in the dendrites, or you can see that in the postsynaptic membrane. Then is the mechanically gated channels, and that is mainly in the skin, but you can also see it in other areas of the body as well, for example in the joints. And there is a distortion of the plasma membrane. It has to be physically changed, and when it's changing, it will open up this channel, and then the ion can enter. And thermally gated channels, they are mainly in the skin, but you can also see it in hypothalamus and some other areas of the body. And they are either responding to cold or a hot temperature, and they become active and then allow the ion to enter, and then you have that sensation, you can feel it. What is the depolarization and hyperpolarization? So normally we have this plasma membrane, and that's separating extracellular fluid from intracellular fluid, which is cytosol. If there is no stimulation there, so at resting membrane potential, you have the negative 70 millivolt. Okay, so that's no stimulation. But what if you get a stimulation somewhere here? So here you got a stimulation. A chemical which is a neurotransmitter attaches to the ion channel. This is a ligand gated channel, and sodium, for example, or could be calcium, enter the cell. They are positively charged, and what happens that it was negative 70 millivolt, and now it's negative 60. And again, it's not the exact number. It can be negative 55. It can be different numbers, negative 40. So so this is becoming less negative now, less negative or more positive. Why? Because it is going towards zero, so it's getting more positive, and that is called a depolarization. So anytime that there is a change in the resting membrane potential, that inside the cell become more positive, that is depolarization. But look at a different situation here. So again, we have the extracellular fluid, intracellular fluid, you have the plasma membrane, and you have these gated channels. So it depends on the type of the neurotransmitter that is released. Now the positive charges inside the cell, which is the potassium, leaving the cell. Normally at rest it was negative 17. Now inside the cell become more negative. Why more negative? Because the positive charge is left, and the proteins that were negatively charged, they are staying there, so they cannot leave, and that's why it's more negative. The other option is that you have ions, negative ions that can enter the cell. So here in this case, like chloride is entering the cell, making the cell more negative. So if the inside the cell become more negative, farther away from the resting membrane potential, farther away from zero, that is hyperpolarization. So here is the action potential. So you don't need to read all of this, it's very simple. So again, it is the resting membrane potential. Here is written negative 64 millivolt. It can be negative 70, negative 75. Okay, depends on the tissue. Negative 60. Once the stimulus comes here, the sodium channels open up, and the sodium entering the cell, and it has to have a threshold here. It's not shown here in your textbook, but it has to have a threshold. Once it goes above the threshold, then you will get an action potential. So this is very, very important. That's why it's called all or none. So if it's not going above the threshold, that is still a depolarization, but it's not an action potential. So above that, it goes there, and it changes. It will be like a 90 or 100 millivolt difference. Okay, so here is the sodium is coming, and the cell is becoming more positive, and that is the depolarization area. And then the sodium channels stop or blocked, and then the potassium channel is open, and here the potassium is leaving the cell, and the cell becoming more negative because you are losing positive charges here. and here is the hyperpolarization. Why is hyperpolarization? Because that's below resting membrane potential. So this is hyperpolarization, and then it goes back to the resting membrane potential. So during this movement of the ions, you have two different currents. Okay, one is called the active current, the other one is called the passive one. For the active one, when you have the stimulation, the ion channel opens up. Sodium enters the cell, and that is active. It requires an ATP. But here the passive one is that from proximal to the distal. When I'm talking about proximal, it means closer to the cell body. Distal means further away from the cell body. Then the ions are moving further away, and inside the cell or the cytoplasm in this area now changes its current. Okay, and then this one has to activate another ion channel, so this can continue. But here you do not need an extra ATP to move the ions along. So action potential is divided into the saltatory conduction and continuous conduction. In the saltatory conduction, the axons are myelinated. So this is the myelination that you can see. So here you have the voltage gated channel, for example, and the sodium enters the cell. Now the sodium tries to leave, but it cannot leave the cell because you do not have sodium potassium pump here, and here is the myelination. So it has to jump from here all the way to here, so from one area to another area. So this is the myelination is important. So once it jumps from one area to another area, takes very little time, then it goes to this area. So that's the node of Ranvier that we talked about, an area that is not myelinated. And here is that you have another sodium channel, and that sodium channel is a voltage gated channel, okay. Then this become activated. Sodiums are entering the cell, cannot leave the cell, then has to jump to the next node of Ranvier. In the next node of Ranvier, you have another sodium channel. So the movement is very rapid. So the electrical activity is jumping from one to another, like a frog. So that is the saltatory conduction. So it means that the myelinated ones conduction is much faster. The other one is the continuous one. So these are unmyelinated axons. So here you have the gated channel here, and the ions are entering the cell, inside the cell becoming positive, but there is no myelination, so there is no insulation, and the charges will leave, so nothing will happen. So this will be a graded potential, okay, not an action potential. And the difference between graded and action, one of the differences is that the graded potential is localized. It will not go to long distance, so it decays. The action potential does not decay, so it can go to long distances. So you have to have another channel here. Okay, now you have another channel very close to this one. So this one is a voltage gated channel. This one open up, then it will go to this area. Now the sodiums are leaving the cell again, and you have to have another gated channel here, and voltage gated. It should be a voltage gated. Okay, this one open up, and then it goes to here, very short distance. You have to have another one, and you have to have another one, you have to have another one. So the problem with this one is that it has to activate multiple gated channels, and that takes time. So each one takes milliseconds, but still time is time. This is why the conduction on this one is slower. So in myelinated one, conduction is a lot faster. They are usually related to somatic function. You become consciously aware, and that's important for survival. And the unmyelinated ones are the ones that are usually in the visceral organs, and at a subconscious level the conduction one is slower. So here we have the role of the velocity. So how fast ion can move during action potential. It depends on the active current, and it depends on the passive current. So we discussed this before. The active one is the one that the ion channels open up and the sodium enters, and the passive one is that the cytoplasm, is moving the ions distally. So the velocity depends on the two factors. One is the resistance, and from the physics, hopefully you remember it, that depends on the diameter. So if the diameter is small, there is more resistance. If the diameter is bigger, so it's less resistance. So which means that the nerves that are thicker, they can conduct electrical activity faster. The other one is the capacitance. So what is the capacitance? Before the capacitance, let's talk about what is capacitor. So capacitor is that you have the two regions that conduct electrical activities, so it can be outside or inside the cell, and then you have in the middle you have an insulator. So in the case of neuron, the insulator is the plasma membrane, and the two regions are extracellular fluid and the cytosol. So in the capacitance, the ions are accumulating on one side of the plasma membrane. So for example, when during an action potential, inside of the cell has a lot of positive charges and outside of the cell has a lot of negative charges. But the problem is that this is not very good for the action potential. So the best way to do this, you have to insulate this area. When you insulate this area, because there are so many charges are there and they're repelling each other, if you insulate this area, then that will increase conduction. So to increase the velocity of an action potential, what you have to do, you decrease resistance, okay, by having a larger diameter axons, and also you reduce capacitance by myelinating the axon. Then is the refractory period. So what is the refractory period? Okay, so in the refractory period we have to start from here, that is at rest. It is negative 70, nothing is going on. You have a stimulus here. So once you have a stimulus here, then that will cause depolarization, which means the inside the cell become more positive, going towards zero. But it has to go above the threshold. Again, I'm telling you, for action potential, this is really important. So it goes above that, it's all or none. Below that, there is still depolarization will happen, but not an action potential. So here is the sodium channels are open, it will go up, up, up. So sodiums are entering the cell becoming more positive, right? And then okay, so I don't have the other image here that I showed you in the lecture, but you have the two different gates here for the sodium channel. Okay, so you have the activation gate and you have the inactivation gate. So when a nerve is stimulated here, the sodium channel is open up. So this is the gate, the activation gate that opens up these ones, and the sodium enters. So all the way to here, the activation gate is open. But once it reaches here, the inactivation gate now goes here and it closes the sodium channel. See, this one goes here and closes. So that is the inactivation gate closes. So no more sodium can enter. And starting here, the potassium channel now opens. So this is the activation gate of the potassium channel. It opens up. Now the potassium is leaving. So inside the cell becoming more negative. So from here to here, that is called the absolute refractory period. Why is that? Because here the activation gate is still open. So even if you have another stimulus, nothing will happen. The sodium is already entering the cell. But once you have the inactivation gate blocking the sodium channel, even if you have another stimulus, nothing will happen because sodium cannot enter. So this is why it's called absolute refractory period. So then you have the relative refractory period, and the relative refractory period, the sodium channel goes back to the resting state, which is the activation gate is closing, the sodium channel and inactivation gate is still open. So there's no more sodium is entering, right? But the potassium channel is still open. So more potassium is leaving, and now is becoming the resting membrane potential, so reaching there, but it will not stop there. It will continue going down. It becoming more negative. So this is the hyperpolarization that we were talking about until the potassium channel closes, and eventually it reaches back to the resting membrane potential. So here is this area is called the relative refractory period, and the reason for that is that you have if you have a large stimulus, then you can get another action potential, so you can get another one. But if you have a small one or a regular one like this one, you will not get anything because the cell is more negative. Once the cell is more negative, you need a stronger stimulus, otherwise nothing will happen. So the synaptic transmission, you have two types. One is electrical synapse, the other one is the chemical synapse. You have here you have one neuron, and this is the axon terminal. So this area is called the presynaptic membrane, and this is the postsynaptic membrane. So this is one cell, this is the presynaptic membrane here, and this is another cell, and this is the another cell membrane. Okay, so the synapse here, it may not work, but I just want to compare this with this one because this is the actual real synapse, and this is just the intercellular space between the two cell membranes. Okay, so these two cell membranes are connected by these proteins. Okay, these are the proteins. These proteins are called connexons. So there is a tunnel there that's open, and ions and small molecules can pass through it. And the small molecules are a different issue, but here we are talking about the ions. So for example, sodium can pass from one cell to another cell. So the neighboring cell can be activated, the neighboring cells. So this is really important for cardiac function. It's also seen in the hypothalamus and also in the respiratory center and medulla. You see this type of synapse, which is called the gap junction. In reality, it's called a gap junction. Okay, it's not does not have a synaptic cleft. The chemical synapse is more common, a lot more common, and the difference between the chemical synapses that you may or may not get an action potential. An electrical one, I told you, another neighboring cell become active, so it means it's always positive. The electrical synapse also you can see it in the smooth muscles, by the way. And the chemical synapse is in all over the central nervous system, and also connection between the nerves and the skeletal muscle. Okay, so you have the presynaptic membrane here, which is similar to this one, and you have the postsynaptic membrane, which is similar to this one. That you have the synaptic cleft here. There's a space there, which you do not have it here. So this is because this is connected by this connexon proteins. So you have these vesicles, and these vesicles contain neurotransmitters, and through exocytosis the neurotransmitters are released in the synaptic cleft here, in the synaptic cleft. And then you have these channels. So these channels should have receptor for these neurotransmitters. So without the receptors, they cannot become active. Once they become active, the ions enter the cell, for example sodium, and then the postsynaptic membrane become active. So in this case here you have the dendrite, but you can also have the skeletal muscle. So you can have another cell body, or you can have another axon, but most commonly is the dendrite. Okay, that's the most common connection. So this cell depends on the type of the neurotransmitter, depends on the type of ion that enters, may or may not become active, may or may not. So what happens here, you have electrical activity. So there's an action because it is an axon. So the electrical activity coming because the sodium channel opened up. So as we discussed before, now it reaches here. This is the axon terminal, this bulb area, okay, the enlarged area, and they have these calcium channels. Now because of this action potential, the calcium channel will open up, okay, the calcium channels. More calciums are coming from outside of the cell entering the axon terminal, and these calcium are attaching to these vesicles, and that will cause exocytosis of these vesicles. So inside the vesicles you have the neurotransmitters. They're already pre-made and they release here in the synaptic cleft, and you have these receptors here in the postsynaptic membrane. This is the postsynaptic membrane, and you have these receptors. They pick up these ones, and what will happen? They become active, they open up, and more ions enter the cell. So again, depends on the type of neuron. It depends on the type of the ion. So they become active or may not become active. So sodium, it will make it more positive, and chloride it make it more negative. So hyperpolarization or depolarization will happen. But the main comment or the main topic here is this one. So the calcium. So without calcium, what will happen? Nothing. So even if you have an action potential, if the calcium is not there, nothing happens. So level of normal calcium is very important for the body, and hypocalcemia, it will cause weakness. So there's two different mechanisms there. One is the neurological mechanism here, and there is also muscular mechanism that inside the muscle you need to have a lot of calcium for the fibers, the myosin and actin to move. So you need calcium for both. So hypocalcemia will cause severe weakness. Then what will happen here in the postsynaptic membrane? So I told you before, it depends on what type of ion enters. So if a positive ion enters here, then that's called excitatory postsynaptic potential. Excitatory. So sodium or calcium enters, activating this area, the postsynaptic membrane, and this is becoming more positive, so that's excitatory. But if the chloride is entering, okay, so then what will happen inside the cell become more negative. So this is the postsynaptic membrane, and it's farther away from the threshold, farther away from the resting membrane potential, and that is inhibitory postsynaptic potential. You need both of them, okay. Sometimes you have to inhibit things. For example, when you want to go to sleep in the middle of my lecture, you need this kind of inhibitory postsynaptic potentials. Otherwise, activity will continue, or the muscles want to relax. So this is another reason. So you do not always need a stimulation. You also sometimes need inhibition. For the excitatory, so this mechanism applies mainly in excitatory. You have two different other mechanisms, like sub-mechanisms. One is called spatial summation, the other one is called temporal summation. So summation is adding multiple stimuli. So by adding that, it tries to increase the electrical activity or the potential inside the cell, and that can cause an action potential. So in the spatial summation, you have multiple axons that are connected to dendrites of the one neuron, and they are coming at the same time. They are located in different parts of the dendrites, but they are coming at the same time. And once they become the accumulation of the all charges, it will go above the threshold, and that can cause an action potential. Okay, so multiple stimuli at the same time. But the temporal one is the one that is one axon is sending signals back to back to the postsynaptic membrane. And here, if there is a small one, nothing will happen. You have to have many of them back to back, very close to each other, so they can accumulate. Once they accumulate, it goes above the threshold, and you can have an action potential here. So for both of these two conditions, it means that small stimuli will not do anything. You have to have either many stimuli coming at the same time, or stimuli come back to back very close to each other in the same area, and that will cause an action potential. And in real life situations, that's actually happening because majority of the stimuli are very small, so it will not cause anything, and that is good. You do not want to have your nervous system activated by each small stimulus. But if the stimuli are important, it means you have to be aware of it, or something has to be done, and at that subconscious level. So that's why this mechanism is there. So now let's talk about the type of the receptors. So you have the two type of receptors. One is called ionotropic receptors, and here you have the ion channel. This is the plasma membrane. You have a receptor, and neurotransmitter is attached here, and it allows the ions directly to enter the cell. So that is activating the postsynaptic membrane. Okay, so this ionic receptors. The other one is called metabotropic or metabotropic receptors, and there is two types. Here you have a receptor here. This receptor is not the channel, it is a transmembrane protein. It has a receptor, neurotransmitter comes here, and then this receptor is activating a G protein, which is a protein inside the plasma membrane, and then a subunit of the G protein will activate the ion channel, and then the ion can enter the cell. Another one is that you have the same receptor here, the same protein here that has a receptor, and it will activate the G protein, but now the G protein is not activating the ion channel directly like this one. It will go and activate an enzyme, and that enzyme is a protein in the cell membrane. This will go and activate a second messenger system, which is like cyclic AMP or cyclic GMP, or increase the level of calcium inside the cell, and that will activate other enzymes like kinases, or it will go and open up ion channel, and then the ion can enter the cell. So two different type of receptors. So this slide, you do not have to memorize this slide. This is just an overview of different type of neurotransmitters. So majority of them are a small molecule neurotransmitter. They can be excitatory or inhibitory, so I will explain those ones separately. And you have the neuropeptides that will influence the neurotransmitter. So the neuropeptide can be released at the same time, as neurotransmitters can affect them. Okay, so they may be released from presynaptic or postsynaptic, and they have to be made usually compared with the neurotransmitters that's already there and ready to be released. And the effect of the neuropeptides are longer than neurotransmitters. Then you have one gaseous neurotransmitter, which is the nitric oxide, which will cause vasodilation. So let's talk about the neurotransmitter. The very important one is the glutamate. This is the most common excitatory neurotransmitter, important for memory, learning, and motor function. You have both type of receptors. The ionotropic ones, they have the NMDA, and they have the non-NMDA, which is the AMPA and kinase, and they will allow sodium and calcium to enter the cell. Too much of the calcium, stimulation of too much of this type of receptors that responding to glutamate is bad, because it will cause cell damage and it will cause cell death. So that's bad for some diseases, for example a stroke. This release of too much glutamate will cause more cell damage. You also have the metabotropic receptors, and those ones are also excitatory. So both types are excitatory. But remember, for memory and learning, and also for motor function, this is not the only neurotransmitter. There are other neurotransmitters as well. Glycine is an inhibitory neurotransmitter, and it is it attaches to a ionotropic receptor. And GABA is another big one, inhibitory neurotransmitter. So They have GABA A and GABA C, which is inotropic, and GABA B that are metabotropic receptors. So the GABA are important for different mechanisms, and also they can be used for different type of medications. Actually, there's GABA, which I will discuss it in the lecture. Acetylcholine. Okay, so this is another neurotransmitter. You see that similar to glutamate. So it's for memory, learning, and motor function. So in the central nervous system, you have it in the cerebrum and the cerebral cortex, but there's two areas in the brain that is important, has a higher concentration of the acetylcholine. And this area is called the basal nucleus of the main and that is in the forebrain, in the frontal lobe area of the cerebrum. And another one is in the brain stem, so that is in the dorsolateral pontine tegmental area. So this is the pons and this is the midbrain, so this is an area, an upper part of the brain stem. And as you can see, they have a lot of connection in other parts of the brain. So you can see this type of neurotransmitters inside the CNS. You can also see it in the peripheral nervous system, ganglia, and you see that the connection between the two neurons in the peripheral nervous system and in an area that connects the motor neuron with the skeletal muscle. So that's called the neuromuscular junction, a motor neuron with a skeletal muscle. You have two types of receptors. One is called nicotinic. The nicotinic one is the inotropic, and these are the ones that are causing activation. And there you have the muscarinic ones. So the muscarinic ones are the ones that may or may not cause an activation. So may be excitatory or maybe inhibitory. So that depends. And these are important, especially important for the autonomic nervous system. For example, in the neuromuscular one, you have the nicotinic one, but in the autonomic nervous system, you have both of them. Okay, which we will discuss it later on. And you have this enzyme acetylcholine esterase that will metabolizes acetylcholine, and the part of the acetylcholine is taken back by the axon terminal, and then this is how it is recycled. Dopamine is important excuse me, for drive, emotion, motivation, reward, but also motor function. So again you see that motor function depends on multiple neurotransmitters, and this is the metabotropic receptor. It needs metabotropic receptor. It can be excitatory, and they are D1 receptors, or it can be inhibitory, which are the D2 receptors. So dopamine can be excitatory or inhibitory. Norepinephrine, these are excitatory neurotransmitter. They are important for wakefulness. So in the morning when you wake up, that is because norepinephrine is released, but this is not the only neurotransmitter. It is important for attention and focus. So if you focus on this lecture, it means you had a lot of norepinephrine released, but too much of norepinephrine is bad because that will cause stress. And it's important for the motor function, and the motor function here is the autonomic nervous system, especially the sympathetic nervous system is important here. So it has the metabotropic receptors, but they also have alpha and beta. So this is a type of receptors that responding to norepinephrine, but again those receptors are divided. The alpha and beta are subdivided into other type of receptors, and they are located in different part of the organs in the body. So once a norepinephrine is released, it doesn't mean that everything is activated. It depends on the type of receptor in each tissue. So we will come back to this later on in a different chapter and discuss it in more detail, and I will tell you the receptors in each organ. There is also epinephrine. So epinephrine is more much more released from adrenal gland than uh, the end of the axon. So an easy way to remember: norepinephrine, neurotransmitters. Each one is starts with N. Epinephrine is more from adrenal gland, but adrenal gland also release norepinephrine. Histamine is an excitatory receptor, an excitatory neurotransmitter, and they are attached to metabotropic receptors. It's important for wakefulness, so this is another neurotransmitter that's important for wakefulness and attention. Also, they are involved in pain, sexual pleasure, blood pressure, and pituitary function. So a major part of neurons that release histamine is located in the hypothalamus, but they are also located in the limbic system and the basal ganglia or basal nuclei in the brain. So when we get to the hypothalamus, I may remind you there at that time. Serotonin can be excitatory or inhibitory. It is important for circadian rhythm, so it makes you more sleepy. It's important for mood and emotion, and it involved in descending pain pathway. It means that it's reducing pain. Most of them is metabotropic, but it can be ionotropic, which is excitatory. Serotonin is involved in depression, anxiety as well. And ATP can be broken down to adenosine, and that can attach to the same receptor as ATP, and these purinergic receptors can be ionotropic or it can be metabotropic. And ATP are neuromodulators. Okay, so these are the ones that are affecting other neurotransmitters, and they will increase their activity or inhibit their activity. Depends on the type of the neurotransmitter. So they are affecting other neurotransmitters. And the neuropeptide, which is again in your textbook is written under neurotransmitter, but they are actually neuromodulators. And they are involved in pain, so either increase or decrease the pain. So for example, substance P increase the pain, so this P, and there are other ones that are decreasing the pain, like enkephalin, dynorphin, endomorphin. So those are the ones. Endorphin. These are the examples that decrease the pain. And you can see this image. This you do not have this image, so this is not tested in the exam. Just to give you an idea here, so you can see that the serotonin, this is part of the descending pain pathway there. You also see the neuropeptides, enkephalin here and enkephalin here, and they are important also for reducing pain. So just to show you the pathways, but this will not be tested in the exam. And stress, okay, something that you will not experience in this class. Then there is two examples. That one is the CRH, the corticotropin releasing hormone, and that is from hypothalamus, and then is another one is the adrenocorticotropin that are affecting the adrenal gland, and then both of them, and they are releasing norepinephrine and epinephrine. So they're part of the fight and flight response system in the body, which we will discuss it in a different time. Okay, and that's it.